Which of the following event photosynthesis does not require light? I have already discussed with the 7th question, that is reduction of carbon dioxide takes place and hexose sugars are formed. Carbon dioxide is reduced and its result is formation of hexose sugar in the stroma of chloroplast. And for the reduction of carbon dioxide, there is no use of light energy. Light energy is not required directly here. Products of light phases are required, that is ATP and NADPH2. Otherwise, there is no direct involvement of light energy. That's why answer 4 is correct. It is a part of dark phase. That is, it takes place in dark phase of photosynthesis. It takes place in dark phase of photosynthesis. Question number 9. Chlorophyll A with weak absorption, peak absorption 680 nm. Actually, there are two pigment systems in plant. Pigment system 1 and 2. Photosystem, they are also called photosystem 1 and 2. And these photosystems are actually bundles of pigment. 200 chlorophylls, 50 carotenoids, electron carrier and one specific form of chlorophyll A. In pigment system 1, chlorophyll A specific form, that is P700, is there. It is the reaction center of pigment system 1, whereas P680 is reaction center of pigment system number 2. So here the answer is very clear, that is P680. P680 is reaction center of pigment system number 2. These pigment systems are present over thylakoid membrane. If we see the thylakoid structure, thylakoid is membrane bond structure. Pigment system 1 is towards upper side of the membrane, whereas 2 towards lower side. This is location of pigment system 1. This is location of pigment system 2 on thylakoid. One is appressed and other one is epressed. So this is the location of pigment system 1, and these are the one specific pigment of chlorophyll A regarded as P680 and P700. P700 is reaction center of pigment system 1, whereas P680 is reaction center of pigment system number 2. Electron transport chain. It is also located on thylakoid membrane. The location of electron transport chain is thylakoid membrane. ETS is located in the membrane. It is also found in the mitochondria, but it is associated with mitochondrial inner membrane. Whereas in chloroplast, this ETS is located on the membrane of thylakoid. This is thylakoid and this is its membrane. And these electron transport system or electron transport chain is concerned with the membrane of thylakoid. Question number 11. What is the net gain of non-cyclic electron transfer? See this word, non-cyclic electron transfer. It is also called Z scheme of electron transfer. Z scheme. Non-cyclic electron transfer is called Z scheme of electron transport. Second, this was reported by Hill and Bendel. Z scheme of electron transport was reported by Hill and Bendel. And in this electron transfer, pigment system 1 and 2 both are involved. Pigment system 1 and pigment system 2 both are involved, then only non-cyclic electron transport takes place. And this non-cyclic electron transport results in synthesis of NADPH2, ATP as well as oxygen, evolution of oxygen. This is the net gain of non-cyclic electron transport, net gain of non-cyclic electron transport. This is pigment system number 2. It absorbs light energy and due to which electron is released from its reaction center. This electron is released due to photoexcitation of chlorophyll. When PS2 loses electron, it is responsible for the photolysis of water. That is, water undergoes lysis, produces OH negatively charged ion and positively charged H ions. These OH ions are negatively charged. They provide electron to pigment system 2. That is, water compensates the loss of pigment system number 2. Now, these electron, the that is return of these electrons is now not possible. These electrons are now utilized somewhere else because the loss of pigment system 2 is also covered by the photolysis of water. Meanwhile, you see hydrogen ions are available. These hydrogen ions chemi-osmotically released from the thylakoid membrane into the stroma, where they are trapped by NADP, where they are trapped by the NADP. So these are trapped by the NADP, but they are positively charged. One more drawback, that is they are positively charged. So these electrons, that is when PS1 excites, now the role of pigment system 1 is here, that is PS1 on excitation, that is absorption of light by PS1, emission of electron by PS1, this is electron emitted by the pigment system 1. These electrons are immediately utilized by H ions for the formation of NADPH, for the formation of NADPH2. Now, the electrons of 1 is utilized in the formation of NADPH2, then there is a loss of electron, it is fulfilled by this electron, it is fulfilled by the electron of pigment system number 2. So this electron is now taken up by the pigment system number 1. See this type of electron transfer, this is non-cyclic electron transfer, this is non-cyclic electron transfer, and during this electron transfer ATPs are also formed, ATP synthesis also takes place. So one compound ATP, other one NADPH2, and here OH ions, after providing electron to the pigment system 2, they are converted into H2O plus O2. H2O plus O2, and this oxygen is released in the environment. So what are the result? NADPH2 formation, ATP formation and evolution of oxygen. So last answer is correct. 12th question. Splitting of water, I have already described. See this one. Pigment system 2 is responsible for the lysis of water, then only this is the possibility of the formation of NADH2. So pigment system 2, when loses electron, to compensate its loss, it is responsible for the lysis of water. So answer is PS2, pigment system 2, responsible for the lysis of water and evolution of oxygen. Lysis of water and oxygen evolution. That is, water undergoes lysis that is positively charged H ions and negatively charged OH ions are produced, and these OH ions supply their electron to pigment system number 2. Because pigment system number 2 on excitation releases electron, so needs electron, so water donates its electron to pigment system number 2, and here manganese ions, Mn ions and Cl ions are required. Manganese and chloride ions are needed for the transfer of electron of water to pigment system number 2, Mn and Cl ions. One only, if one only, then answer is ATP. Pigment system 1 concerned with cyclic electron transfer and its result is synthesis of ATP only. Whereas third one, when both interact, then there is a possibility of non-cyclic electron transfer, and it is useful in multiple or many ways. That is water ka lysis, lysis of water, second oxygen evolution, third NADPH2 formation, fourth ATP generation. That's why the multiple use when both systems interact, when both system interacts. Now 13th question, membrane or lamellae of grana. Yes, grana lamellae, that is thylakoid restricted to granum only. These are the granule thylakoid. These are the granule thylakoid which are related with the granum only, and they have pigment system 1 and pigment system number 2 both. Then suppose this is neighborhood granum, this is neighborhood granum, it has also granule thylakoid. You see some thylakoids are with extension and they are from one granum to another granum. These are called stromal lamellae. These are known as stromal lamellae or fret. They have only pigment system number 1. So check the answer. Membrane of lamellae of grana has PS1 and PS2 both. That is right answer. First option is right. This is right. Second, stromal lamellae lack PS2. Yes, clearly see, you see here is only 1 is present, that is 2 is not present, and NADP reductase enzyme is not at all concerned with the pigment system number 2. So this is the second one. It is also correct. The non-cyclic photophosphorylation occurs when only light of wavelength beyond 680. Beyond 680 inactivates pigment system 2. So non-cyclic electron transfer is not possible. If the wavelength is beyond 680, in that case only pigment system 1 is effective, and if only 1 is effective, then there is no possibility of non-cyclic photophosphorylation. So the third option is wrong. First two are right. First two are right. Third is wrong. So answer is this one. This is the answer for what? That is first is correct, second is correct, third is wrong. And 14th question, in photosynthesis electron transport system, what is the role of manganese? I have already described. Then there is a lysis of water. There is a lysis of water. Water undergoes lysis, produces H ions and OH ions. These OH ions are negatively charged, they release electron. This electron is taken up by pigment system 2 reaction center, that is P680, and in this process Mn ion and Cl ions are needed. Manganese and chloride ions are needed for the transfer of electron from water to pigment system number 2. So that's why pigment system 2, when it is it is responsible for the lysis of water, so definitely Due to the lysis of water, these OH ions also responsible for the release of oxygen. So PS2 is also known as oxygen evolving complex. Pigment system 2 is also called oxygen evolving complex. Now come to the 15th question. Photolysis of water, that is to check a water molecule H2O. That is, if it splits, then 2H, 2 hydrogen ions are produced. And if 2 hydrogen ions are produced, then 2 electrons are also produced. So 2 photons and 2 electrons. 2 photons and 2 electrons are released. Oxygen produced in the photosynthesis process. It is confirmed by Rubin, Hasid and Kamen. Rubin, Hasid and Kamen. These fellow use 18O containing water. 18O containing water. This is isotope of oxygen. Isotope of oxygen supplied with water. Supplied with water, and they observed evolved oxygen is O18 type. Hence proved that is oxygen is evolved from water, not from CO2 by Rubin, Kamen and Hasid. Then DCMU, CMU direct question. It is inhibitor of pigment system number 2. It is an inhibitor of pigment system number 2, and pigment system number 2 is associated with the oxygen evolution and non-cyclic photophosphorylation. So DCMU, dichlorophenyl dimethylurea. Dichlorophenyl, PHE, phenyl dimethylurea. It is an inhibitor of pigment system number 2. Inhibits photolysis of water and oxygen evolution. If water lysis does not take place, in that case non-cyclic electron transfer is not possible. 18th question. 1.8. In which of the following oxygen does not evolve during photosynthesis? Simple question. That is green algae and blue-green algae. In all these, water is used as raw material and oxygen is evolved in them. Bacteria, they do not use water as electron donor. Bacteria do not use water as electron donor. They use sulfur compound. Sulfur compound or any organic compound as electron donor. So in their photosynthesis, oxygen does not evolve. In their photosynthesis, oxygen does not evolve. That's why the question number 18, its answer is 4. Its answer is 4. That is photosynthetic bacteria. They don't use water and they do not evolve oxygen. Even then, their photosynthesis takes place in infrared light, and they don't have chloroplast. Their photosynthesis takes place in chromatophore. That is, bacterial photosynthesis is termed as non-oxygenic or anoxygenic. Non-oxygenic photosynthesis. Non-oxygenic photosynthesis. Bacterial photosynthesis is non-oxygenic or anoxygenic photosynthesis. 19th question. Which of the following is needed both in photosynthesis and respiration? Direct question again. That is electron transport system. Electron transport system is found in chloroplast as well as mitochondria. Chloroplast electron transport system is involved in the photosynthesis activity, whereas an electron transport system is mitochondria is involved in the respiratory activity. And in both of them, cytochromes are present? Cytochromes are present as electron carrier in both the electron transport system. So my answer for this question is fourth, that is cytochromes are needed in photosynthesis as well as in respiration. They are electron carrier. These are all electron carrier. Iron porphyrin compound. Cytochromes are iron porphyrin compound. Cytochromes are iron porphyrin compound. Then 20th question. 20th question, that is, which of the following contains copper besides iron? Final answer is cytochrome A3, but this is related with the respiration. Cytochrome A3 is found in mitochondrial electron transport system. It has copper as well as iron. It has copper as well as iron. Mitochondrial ETS has mitochondrial electron transport system has cytochrome A3. Then Rubisco. Rubisco is an enzyme. Rubisco is an enzyme, better to say most abundant enzyme at this globe. Most abundant enzyme is at this globe, and its full form is ribulose bisphosphate carboxylase and oxygenase. Ribulose bisphosphate carboxylase and oxygenase. That is C for carboxylase, O for oxygenase. That is, it has dual role. It has dual role. One involve in carboxylation and may involve in oxidation if adequate supply of carbon dioxide is not there. If adequate supply of carbon dioxide is not there, in that case it performs activity of oxygenase also. So Rubisco, its utility, it's a main enzyme. It is a main enzyme responsible for the carboxylation of RUBP. This enzyme is concerned with the carboxylation of RUBP. It is the first step of Kelvin cycle, results in the formation of 3-PGA as first stable product. This reaction is catalyzed by this particular enzyme. So that's why Rubisco enzyme found in all photosynthesizing plants, all the photosynthesizing plant, and it is concerned with the carbon dioxide fixation in the dark phase. Carbon dioxide fixation in dark phase. Then autoradiographic technique. By this technique, use of 14C isotope of carbon. Used by whom? Kelvin. On which plant? Chlorella and Sandesmus. Experimented over chlorella and Sandesmus, and they studied various steps of reduction of carbon dioxide during the formation of hexose sugar, and that cycle is named as Kelvin cycle. So that's why the answer is trace the path of carbon reduction in photosynthesis process. and use the isotope of carbon, that is 14C, 14C or C14. Question number 23. This is three major part of Kelvin cycle. Three major part of Kelvin cycle, that is 1, 2 and 3. Kelvin cycle, see here. Here indication is already given. This carbon dioxide is trapped by ribulose 1,5-biphosphate and 3-phosphoglyceric acid. 3-phosphoglyceric acid is formed as first stable product. First stable product. So part 1 is carboxylation. Part 1 is carboxylation. It can be identified by the incorporation of carbon dioxide. And see this one. NADPH. NADPH. That is ability to supply hydrogen. Hydrogen ions are given to the 3-PGA. Then only hexose sugars are formed. That is, if any reaction hydrogen ion is taken, that is reduction. So part 2 is major part of Kelvin cycle, that is reduction. That is reduction. Then after the formation of hexose sugar, you see this arrow indicates the reformation of RUBP. So last part, that is third part of this Kelvin cycle, is regeneration. Third part of Kelvin cycle is regeneration. That's why the answer is now very clear. That is carboxylation. Second one is reduction. Third one is regeneration. This is display of Kelvin cycle. This is graphic representation of Kelvin cycle in 23rd question. Now come to the next one. First stable product of photosynthesis, that is direct question. 3-phosphoglyceric acid, 3-phosphoglyceric acid, 3-PGA, better to write here 3-PGA. How does it form? That is RUBP, ribulose 1,5-biphosphate, CO2 acceptor. It undergoes carboxylation with the enzyme Robesco. Here Robesco enzyme is required. Reaction 1, carboxylation. First of all, 6-carbon keto acid is formed. It is unstable. This keto acid is unstable. And this keto acid uses water and converted into 3-phosphoglyceric acid 3C compound as first stable product of photosynthesis. 3C compound as first stable product of photosynthesis. And all those plants in which 3-PGA is first stable product of photosynthesis, they are called C3 plants. All these plants are known as C3 plants. So answer is 3-PGA. Whereas in C4 plant, the word C4 already indicates that is 4-carbon compound is its first stable product, and that is oxaloacetic acid. Oxaloacetic acid is first stable product of photosynthesis in C4 plant. How does it form? It is formed by the PEP, phosphoenol pyruvate. When it combines with CO2, it forms oxaloacetic acid in C4 plant. That is, C4 carboxylation starts with the PEP carboxylation, whereas in C3 plant it starts with the RUBP carboxylation. 25. Number of molecules and turns of Kelvin cycle required for the removal of one molecule of glucose. One molecule of glucose, that is 6-carbon compound, C6, H12O6. In that case, for 6C, 6 carbon dioxide are needed, 6 CO2 are required. For 1 CO2, 1 Kelvin cycle. For 1 CO2, 1 Kelvin cycle. That's why for 6 CO2, 6 turns of Kelvin cycle takes place and 6 molecules of carbon dioxide are utilized. This is the answer. This is the correct answer. Already marked actually, sorry.